Welcome friends. Today we will continue with the discussion of fluorescence microscopy. Last day we have just done the overview of fluorescence microscopy we did a little about epifluorescence microscopy. So today let's start from the revision part what we did in the last class. So what is fluorescence microscopy? So these microscopes, they produce an image from the light that passes through the specimen. So if I consider that this is the specimen, say for example few molecules which absorb the light. So these molecules, they absorb the light and they absorb, say for example, they absorb the radiant energy. These molecules, then they become excited. Okay, these molecules become excited. After these molecules become excited, they will release their trapped energy as light. So I write here, they release their. Okay, so these excited molecules will be releasing their trapped energy in the form of light. We all know that the light emitted by the excited molecule will have longer wavelength. The light which is emitted by the excited molecule, this wavelength will be longer than the wavelength it has absorbed. Clear? Now this fluorescent light, it is emitted very quickly by the excited molecule, as it is giving up the trapped energy and it is returning back to the stable form. Am I able to make you understand? I said here the specimen will absorb the light and the specimen will become excited, and this after they have excited, they will release their trapped energy in the form of light. The light which they emit has longer wavelength than it has absorbed. They immediately give the light as they become quickly excited, quickly release the energy in the form of light and then become stable. That is why the emitted light will be very quickly seen. Now another important thing to note down that these fluorescence microscope, they exposes the specimen to which kind of radiant light? So they exposes the specimen, just think, they exposes the specimen to which kind of radiant light? It is UV, it is violet and it is blue light. Okay, it is UV, violet and blue light. These are the lights which excite the molecule, which is used in fluorescence microscopy. Okay, now here the light source is usually a high pressure mercury or xenon vapor lamp, and more recently lasers and LED sources are used. Clear? So here the light source, what is the light source? Here in fluorescence microscopy, it can be mercury, xenon, vapor lamp, and more recently the LED sources have been used. Clear? Now this is a very interesting picture where there is a comparison between the bright field and the dark field. Bright field and this is a dark field. Okay, images. So here what we see that, so again this one is the bright image and this one bright field and this one is dark field. So what we see here, this is the picture of the Drosophila. What we see here in this picture, that the sensory bristles which you can see here, which is dark in color here, this appears on the surface of the fly. They appear dark in color on a white background. Okay, so in the bright field microscope, the object will be seen in darker color while the background is white in color. Okay, this is the bright field image. And what we see here is that I change the color. And it is seen as white on a black background. It is seen as white. Understood? It seems white on a black background. Now in this picture number C and D, what we can see, this is the picture of phase contrast image, and what we can see here is that these cells are grown in the tissue culture, and here what we can see that these two images, they are extracted from a time-lapse video sequence at different intervals, and we can see that there is a phase halo in this picture, which is visible in the phase contrast microscope. And this picture is of differential interference contrast. In short, we say it as DIC, differential interference contrast image. In short, we say it as DIC, and this is the image of Volvox. Okay, here what we say that the image of two focal planes of the multicellular alga Volvox is seen here. Now see this picture. This picture is a comparison of epifluorescence and confocal fluorescence microscopy. So see this epifluorescence microscopy, it is a conventional epifluorescence microscopy where the resolution is very low. We cannot see the fine details in this picture using the conventional epifluorescent light microscope. In second and third, they used laser scanning confocal microscopy, which is the latest one. Here the fine details of the microtubules can be seen, and also we can see that this B and C, they are the resolution of two different resolution settings. What we can see here that they are the representation of two different resolution settings of this confocal microscope. So this image B, it was collected with the pinhole which was set to a wider aperture than C. Okay, so this we can conclude that confocal microscopy gives better resolution than the conventional epifluorescent light microscope. Okay.